This is our final presentation, presented by Assad Ansari, Mackenzie Reed, Ali Higgins, and James Matson. So we live in Raleigh, which gives us the benefit of having a lot of trees around. Even though we're not considered a rural area, we do have a lot of opportunity to see nature. So because of that, we're able to see what kind of damage trees have had throughout their lifetime, like through herbivorous damage and fungi. And we've managed to notice, at least in our data, that there was chewing, mining, and scraping. And the fungal damage that we did manage to observe was mostly lichen. However, the purpose of our experiment was to record a bunch of different types of damage on a bunch of different trees and gather an understanding of why the damage differs. What we did was we got a size of 8 different tree leaves and their damage and the size of their damage on each leaf, and compared it to the environments the trees were located in. For example, if it was in a congested area or non-congested area, and how we defined that was a congested area was one that was more urbanized and had a lot more human interaction in it. And a non-congested area was an area with a lot less. So for example, a forest, a lake, a park. And what we had hypothesized was that tree leaves would have a lot more damage in non-congested areas. And what we were able to see was if that was true or not, and if human interaction actually caused a lot more damage to leaves or a lot less. So for our methods,
So the Leaf Herbivory Ecology Lab came to us in 4 separate parts to ensure proper understanding of the subject. We were the first part, we were tasked with going outside and collecting data from 10 leaves for 2 separate species each. And we used that using a 1 centimeter grid paper. And after doing that, we measured the surface area of herbivory or fungal damage on each of the leaves. This included fungal damage, mining, scraping, and chewing. After that, each group member took their own data and they made their own qualitative and quantitative data graphs. After that, all the group members came together, and with the data of 8 trees, they made a group quantitative and qualitative graphs. Having an increased number of data from 8 trees helped us get a better understanding on leaf herbivory and how it affects different trees in different environments. For our quantitative data collection, we used a centimeter squared grid paper to measure 10 leaves on 8 different trees and their damage. For our qualitative data, we observed the surrounding environment to analyze if it was considered congested versus non-congested. How the measurements were calculated, we gathered the data of each leaf on the 8 different trees and found the average size and damage. This information was gathered and put into a bar chart shown in Figure 1. And the environment was classified. We looked around the trees, and if there was a sidewalk or a building within 20 feet of each tree, we considered it a congested area. If the tree had no buildings or sidewalks within 20 feet, we considered it a non-congested area. And on to our results.
So for our results, we gathered all of our data and we put it into a bar chart. So for Figure 1, our data table illustrates that the average leaf surface area of each tree with a relative comparison to the surface area of the herbivory and damage done to each leaf. The quantitative data in this chart is represented by the measurements of the leaves and their damage. Each of these measurements was taken in centimeters squared. The qualitative data that can be taken from Figure 1 would be the pattern of the larger leaf having a larger amount of damage. As it can be seen in Figure 1, Tree 6, the red maple, has the largest average surface area with 74.37 centimeters squared. Relative to this, it had the second highest average surface area of damage with 8.11. Tree 4, the Magnolia denuta, was the tree with the second highest average surface area with 45.3 centimeters squared, and the damage was 9 centimeters squared. Looking into the other trees in the data spread, trees 1 and 2 both had none or close to no amount of damage observed. Tree 7 was the same species as Tree 6, the red maple, had a fraction of the surface area and a very small average amount of herbivory. On average, the leaves with a higher average surface area had a higher surface area of damage. So this is our chart, and this was our data table showing all the numbers of the surface area of the leaf and the damage of the leaf. So here are pictures. As you can see, in this picture you can see the herbivory, in that picture it's also the herbivory, and in this picture you can observe the fungi and the herbivory damage on the leaves. So here's figure 2. In figure 2 we compared the damage of the leaves in congested areas versus non-congested areas. Trees in non-congested areas had significantly more damage than trees in congested areas. The quantitative data is the average measurements of the leaves along with their damage in centimeters squared. The qualitative data in figure 2 is how the environment can affect how much damage the leaves have. Here's our chart, and then again, here's our data table showing all the numbers in the trees. In figure 2, the trees in congested areas had lower amounts of damage compared to trees in non-congested areas. So this was considered a congested area because it was within 20 feet of a building and a sidewalk. And then this was a non-congested area because it was located near a lake with no human interaction.
Next, we have the discussion. The collected data shows how much fungal and herbivore damage is observed on each tree, also shown in figure 2. The trees that are located in congested and more urbanized areas are shown in blue. Trees in non-congested areas are shown in red. The quantitative data includes the measurements of the damage done to each tree. The numbers show the damage of tree leaves in congested areas along with the measurements of leaf damage in non-congested areas, also seen in figure 2. The Lagerstroemia indica, the Franciscanus pennsylvanica, the red maple, and the crepe myrtle were all located in congested and urban areas. They are seen to have little damage. The interference of humans and infrastructure caused the fungal and herbivore damage to decline, compared to the damage on the leaves in the less congested area. This is proven because of studies show that fungal abundance and diversity were notably higher in samples from natural than urban habitats in both the air and the soil. ISME Journal 2020. Studies show that urbanization can also lead to a decreased abundance and diversity of Photophilagos arthropods, Rarup and Shrewsbury and Herms 2010. Both of these findings conclude that the disruption of the habitats in the congested and urbanized areas can affect how species interact with the trees in those areas. It can lead to a decline in both fungal damage and herbivore damage in both those areas compared to the non-congested areas. And next we have our references from 2 different sources, and
then that's it.
it.
Thank you very
Thank
much,
you.
Jonah. It was nice having you as a teacher, and that's 8 minutes.